Hello friends, welcome to a new lecture today. In this, we are going to learn some important points about the amino acid phenylalanine and tyrosine. So, first I will draw the structure of phenylalanine. Phenylalanine and tyrosine, both these are aromatic amino acid. So it contains a benzene group with an amino acid group attached to it. So this is phenylalanine. Whereas tyrosine is to the benzene group we have a methyl group attached to this. The normal structure of amino acid is attached. That means a methyl benzene is attached to the alpha carbon of an amino acid. This is same. It is phenylalanine. Tyrosine is hydroxyphenylalanine. That means for this phenylalanine, if you add a hydroxyl group, then we call it as tyrosine. So this is tyrosine. So if you see the synthesis of phenylalanine, in the synthesis of phenylalanine is synthesized. Phenylalanine is an essential amino acid. This phenylalanine is an essential amino acid. It is not synthesized in our body. We will have to get it through diet. Whereas tyrosine is non-essential amino acid. Tyrosine is non-essential amino acid as it is synthesized from phenylalanine. Phenylalanine, in the presence of the enzyme phenylalanine hydroxylase, phenylalanine forms tyrosine. During this process, tetrahydrobiopterin gets converted to dihydrobiopterin. Tetrahydrobiopterin gets converted to dihydrobiopterin during the conversion of phenylalanine to tyrosine. And this dihydrobiopterin gets converted to tetrahydrobiopterin. That means tetrahydrobiopterin is restored with the help of the enzyme dihydrobiopterin reductase. This dihydrobiopterin reductase converts NADPH plus H plus to NADP. It converts NADPH plus H plus to NADP. Dihydrobiopterin gets converted to tetrahydrobiopterin in the presence of the enzyme dihydrobiopterin reductase. And during this conversion, NADPH plus H plus gets converted to NADPH. NADP, yes. And this dihydrobiopterin gets converted to GTP. And the conversion of, tetrahydrobiopterin gets converted to GTP. This conversion of tetrahydrobiopterin to GTP occurs in the presence of GTP cyclohydrolase. Okay, this pathway is very important. Phenylalanine, which is an essential amino acid, is obtained from diet. This phenylalanine, in the presence of phenylalanine hydroxylase, it forms tyrosine. Tyrosine is a non-essential amino acid, and during this process, tetrahydrobiopterin gets converted to dihydrobiopterin. Now, tetrahydrobiopterin is restored from dihydrobiopterin with the help of the enzyme dihydrobiopterin reductase. And during this process, NADPH plus H plus gets converted to NADP plus. Then, tetrahydrobiopterin gets converted to GTP in the presence of GTP cyclohydrolase. Now, once we have seen this, we will see the special products. Okay, in the special products, the product tyrosine, the amino acid tyrosine gets converted to 3,4-dihydroxyphenylalanine in the presence of the enzyme tyrosinase. In the presence of the enzyme tyrosinase, tyrosine gets converted to dihydroxyphenylalanine. This dihydroxyphenylalanine gets converted to dopaquinone in the presence of tyrosinase. And this dopaquinone has two pathways. One, it forms melanin, that is melanin red, which is also called as pheomelanin. And it also forms through a series of reactions, it also forms melanin black, also called as eumelanin. Right? So, if you see tyrosine, gets converted to 3,4-dihydroxyphenylalanine in the presence of the enzyme tyrosinase, which is dependent on cofactor copper. And during this process, oxygen gets converted to water. And 3,4-dihydroxyphenylalanine also gets converted to dopaquinone in the presence of the enzyme tyrosinase. And even here, there is conversion of oxygen to water. Right. This occurs a series of reactions. Though you do not remember all these, the only thing that you have to remember is tyrosine is responsible for formation of this melanin that gives color to our body. Then here, this dopaquinone to melanin black, there are many intermediates. You do not need to know these intermediates, but I am just telling it for information. So, dopaquinone gets converted to leukodopachrome, and this leukodopachrome gets converted to 5,6-dihydroxyindole, and this 5,6-dihydroxyindole forms indole 5,6-quinone. This indole 5,6-quinone will form melanochrome, which finally forms melanin black. You don't remember these, only if you are done with everything, then try to remember this, or else not necessary. At least remember that dopaquinone, by a series of reactions, it forms melanin black, eumelanin, and melanin red and pheomelanin, and thus the melanin or color of our faces or our body is due to the amino acid tyrosine. Then, next, we will see the next special product. There are two more special products which are important. See, the amino acid tyrosine gets converted to dihydroxyphenylalanine in the presence of the enzyme tyrosine hydroxylase. Tyrosine hydroxylase converts tyrosine to dihydroxyphenylalanine. This dihydroxyphenylalanine, through decarboxylation, it forms dopamine. Dopamine, through hydroxylation, it forms norepinephrine. This norepinephrine undergoes methyltransferase and it forms epinephrine. Right. So let us see the enzymes which are seen. This is important actually. So tyrosine gets converted to dihydroxyphenylalanine in the presence of the enzyme tyrosine hydroxylase. And this tyrosine hydroxylase is actually the rate limiting enzyme. Then dihydroxyphenylalanine gets converted to dopamine in the presence of the enzyme dopa decarboxylase. So as the name says, here carbon dioxide is removed. This dihydroxyphenylalanine can be abbreviated as DOPA. Right? Next, this dihydroxyphenylalanine or DOPA decarboxylase enzyme requires pyridoxal phosphate as coenzyme. Then dopamine is the first catecholamine to be formed in this reaction, and this dopamine gets converted to norepinephrine in the presence of the enzyme dopamine beta hydroxylase. This dopamine beta hydroxylase requires vitamin C, and during this process oxygen gets converted to water. So dopamine, in the presence of dopamine beta hydroxylase, it forms norepinephrine. Dopamine is the first catecholamine to be formed. Norepinephrine, in the presence of the enzyme phenylethanolamine N-methyltransferase, it forms epinephrine. Now, during this process, S-adenosyl-methionine gets converted to S-adenosyl homocysteine. Right. Remembering these enzymes may be difficult, but you should remember this enzyme, which is very important. Tyrosine hydroxylase is the rate limiting enzyme, and this converts tyrosine to dihydroxyphenylalanine. Then this dihydroxyphenylalanine, in the presence of DOPA decarboxylase, it forms dopamine. So you should remember the first reaction is hydroxylation reaction. Then by decarboxylation, the DOPA forms dopamine. Dopamine by hydroxylation again forms norepinephrine. Then norepinephrine by methylation forms epinephrine. At least if you remember these, you can guess the enzymes through the options. Right? So this is the special product, that is catecholamines are synthesized from tyrosine. Then next product, tyrosine is present in thyroglobulin. Okay, in the thyroglobulin, tyrosine to this iodine is added, and if one iodine is added, it forms monoiodine. It is monoiodotyrosine. Whereas if two iodine are added, it forms diiodotyrosine. Okay. If monoiodotyrosine combines with monoiodotyrosine combines with diiodotyrosine to form T3. Whereas diiodotyrosine combines with one more diiodotyrosine to form T4. So T3 contains 3 iodine atoms, whereas T4 contains 4 iodine atoms. Right. The last important topic of this is, look at this. See. Right. Tyrosine gets converted to dihydroxyphenylalanine. See, tyrosine gets converted to dihydroxyphenylalanine in the presence of the enzyme tyrosinase. And this is the process of melanin formation. Right? Similarly, if you see here, even here, the tyrosine gets converted to dihydroxyphenylalanine, and here the enzyme is tyrosine hydroxylase, and this is epinephrine or catecholamine synthesis. Then what is the difference? See, in both these reactions, tyrosine is getting converted to dihydroxyphenylalanine. Okay, but for this conversion of tyrosine to dihydroxyphenylalanine, we have two different enzymes. That is tyrosinase, which is in melanin synthesis. Here we have tyrosinase enzyme, whereas here in the catecholamine synthesis we have tyrosine hydroxylase enzyme. Now the question is, which enzyme acts when? Right? See, this tyrosinase is mainly expressed in melanocytes. Tyrosinase is present only in melanocytes, whereas tyrosine hydroxylase is expressed in sites of catecholamine synthesis. In sites of catecholamine synthesis, like in sympathetic ganglion and nerve endings, okay, in adrenal medulla where catecholamines are synthesized, in all those areas tyrosine hydroxylase is present. Right. This tyrosinase and tyrosine hydroxylase, both these are monooxygenases. But tyrosinase requires copper as the coenzyme, whereas cofactor. Tyrosinase requires copper as cofactor, whereas tyrosine hydroxylase requires tetrahydrobiopterin as the cofactor. So this is the difference of tyrosinase and tyrosine hydroxylase. Catecholamine synthesis does not occur in the melanocytes, because in the melanocytes the enzyme tyrosine hydroxylase is absent. Tyrosinase enzyme is present in melanocytes, and thus in melanocytes tyrosinase enzyme acts, and that will lead to production of melanin. So these are the special products from tyrosine and synthesis of tyrosine. Thank you guys for watching my lecture. Thank you.